Hi there, biochem students. This is Dr. BP checking back in to say, here are the answers for your PCR primer homework number 2. So what I've done is I've annotated this gene. It's sort of color-coded now, so my apologies if this is hard to see. I've annotated it for each primer pair, and I realized that I actually had a mistake in my second PCR primer pair, so apologies for that. This document is available to you for you to peruse as you wish. Um, Email me if you have questions after watching this video and looking carefully at the primer pairs. So primers for number 1 were okay, but I think number 2 had some problems in the reverse primer. So the way these works is if it's red, that means you're looking for the primer pairs that have the forward that looks like that. And so the forward one will actually show. I'm going to see if I can do this in the same colors. The forward does go in this direction, in the 5' to 3' direction here. So it matches. It matches the one here, 5' to 3', ATG, TCT, AGT, right? ATG, TCT, etc. And then the reverse primer that goes over here is going in the opposite direction. So here we want the reverse primer, we want the polymerase to go that direction. And because this gene has the 3' end here, we want the primer to be in the 5' to 3' direction going that way. So when you look at the base pairs here, it should read 5' T, C, A, T. So the reverse primer should be 5' TCAT and then goes da 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 da. So when you're looking at your primer pairs, it's important. The way that I checked yours was just to look at the 5' and then I looked at the first 6 or so nucleotides to make sure that they matched. So yours might not be exactly the same as mine, right? If you used a little bit more or a little bit less, you'll know because your melting temperature will be a little bit different. As long as they're within 1 degree of each other, like 55.5, 54.8, those are fine. As long as yours are in that same range, that's totally fine. It doesn't matter if yours is a little bit different on the end. But the way for you to check is to check the 5' nucleotides to make sure that they match. So the 5' forward should be ATG, ATG, TCT, ATG, TCT. The reverse, going the opposite direction, I'm pairing with this region that I've highlighted, but it's going to be the reverse complement. So this one goes 5' to 3'. So my reverse complement is going to go 5' to 3' this direction, base pairing with these. So 5' T, C, A, T. 5' T, C, A, T. Let's look at the second primer pair. So because it also starts with nucleotide 1, that's the same primer as this gene here, but it's just going to 245. Some of you had some trouble counting with these ones because it's a little bit wacky in how we do this. So let's look at where the orange one is, 245. Notice that, so here's my orange one here. The last one, so you want it to amplify the sequence from 241, 2, 3, 4, 5 to 245. So we want it to start there and then go back in so that just this segment is being amplified. So this is where my primer is going to start. So because this again is a 3' end of this, I'm going to make the 5' end and it's going to pair with the C. So I'm going to make my 5', 5' G, T, G, T, T. 5' G, T, G, T, T. 5' G, T, G, T, T. And then it continues on the next line. T, A, C, A. T, A, C, A. T, A, C, A. So again, make sure that the first part of yours lines up with my answer key. I'm sorry that I did this wrong the first time. So go back and double check. The same thing works for these ones. I'm going to do one of these with you, and then you can go back through and check to make sure yours match. So my primer pair for 325 through 1163. My 325 yellow one should start here with ACT. So let's go up and find 325. It's in yellow. Here's my yellow. This is 301. This is 311. This is 321, 22, 23, 24. 325 starts with the A, right? 325, yeah, 325. So 325 starts here, and I just copied and pasted this into my oligo calc to get the melting temperature. So it should read, let's make this one a yellow one going this direction. It should read 5 prime ACTGAT, 5 prime ACTGAT. So you're looking so that it matches and that it should be pretty close to this whole thing if your melting temperature is close to mine. The reverse ones are where most people had trouble, so let's take a look. We want to start at 1163 and go back into the gene. So this is 1141, 1151, 1161, 62, 63, 1163, double check, yep, 1163. So 1163, we want to go backwards and go into the gene so that it's amplifying just this segment here. That's the size PCR product that you're going to get from this one. So remember, though, that we want it to be the reverse complement of this sequence so that our polymerase can go 5 prime to 3 prime this direction. So that means as I'm looking at the nucleotides I need, I want it to be 5 prime to base pair with this 3 prime. So it should be 5 prime TAAAG, 5 prime TAAAG, 5 prime TAAAG, blah, 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 blah. Again, I didn't check the ends of these. If you got the first 3 to 6 correct, that was where I stopped paying attention. I'm looking to be at around that length so that your melting temperatures match. But this is what I'm looking for. So go ahead and take a look at this to see if it makes sense, and email me if you have questions or if you think I've made a mistake somewhere else, which is totally possible. Good luck, everybody. This is hard stuff. I didn't do this until I was in graduate school. So if you're doing it now and thinking about a career in biomedical stuff, you are ahead of the game. So good luck. Email me your questions.